No, Chairman, distinguished colleagues, ladies and gentlemen. I'm Dr. Solomon Umukoro from University of Ibadan. Briefly, I'll be discussing on Juberlin reduces aggressive behaviors in animal models, predictive of human aggression. Thank you. First, we'd like to ask ourselves the important question, what is aggression and how relevant it is to us? Aggression is nothing but a series of deliberate violent actions that result in harm, injury, or death to another organism. Apart from that, it may also lead to profound long-term effects on the victims of such incidents. For example, rape, etc., etc. Aggression is the common factor responsible for violent crimes worldwide. Apart from being destructive, it may also serve a useful defensive mechanism of self-preservation in life-threatening situations. Several factors may trigger aggression. Apart from neuropsychiatric disorders, the next important factors are what we refer to as traumatic stress, or what we call life events. Everything about stress, which could be poverty-induced, unemployment, loss of beloved ones, etc. Stress has been known to result in frustration and depression, and then act of aggression. Therefore, aggression may be indication of prolonged stress and represent a window for venting aggression on people. Apart from stress, psychiatric disorders, drug abuse, child abuse, and exposure to violent games, as we all are aware today, are among factors that may promote aggression. Aggression is a disorder with well-defined neurochemical abnormalities, principally neurochemicals that are involved in mediating or regulating our moods, behavior, and emotions, like serotonin, dopamine, adrenaline, are involved in aggression. However, serotonin has been found to play a key role in initiating and propagating aggression. So that is what we known as serotonin deficiency of aggression. So an hypothesis stating that aggression is due to low level of serotonin in the brain. And that has been linked up with several evidence. For example, low metabolites of serotonin have been found in the brain of aggressive individuals. And low level of serotonin have been linked to other behavioral disorders. Also, by lowering brain level of serotonin, using drugs and other substances have been found to result in aggression. Conversely, by increasing brain level of serotonin of 580, also reduces aggression, which actually points to the fact that serotonin is a very important chemical that mediates aggressive tendency. All along, attempts to treat aggression started in the early 70s with the use of lithium carbonate, and it was shown in clinical studies to reduce aggressive behavior, perhaps because of its mood stabilizing effect. The mode of actions was ascribed to enhancement of serotonin level, or perhaps reducing adrenergic nervous system activities. That is through adrenaline reduction, which has also been shown to involve in aggression. However, current approach to treatment of aggression involve the use of drugs like haloperidol, which are the mainstay of drugs for treating aggressive behavior and violent tendencies, especially in psychiatric disorder like schizophrenia. These drugs act by through sedation that other thoughts that may trigger aggression, but they have several setbacks. What are the limitations? They impair motor performance, defense, or flight ability of the organism. So in case that necessitate the organism to fight back or to run, all these kind of drugs will impair that mechanism, and that shows that they are not actually good drugs for aggression. Other commonly used drugs are the GABA A agonists. For example, diazepam, nitrazepam, phenobarbital. They also reduce aggression at very high doses, and that by causing sedation, which is also a very major side effect because this drug causes muscle relaxation, and that will impair defense mechanism and flight capabilities. So that set us into thinking that what should be the ideal drugs that we can use to fight aggression. So current effort now is to develop safer drugs that can selectively reduce aggression without impairing other behavioral functions. And the current effort is to search for natural products, that is of plant origins, that are believed to be more safer, as we have heard all along, and easily accessible to the individuals, that could be developed as remedies for aggression. Hence, we look at this product called Jubeline. Jubeline is a commercial product that has gained worldwide recognition as a remedy for anemia and joint pains. Also been said to boost the immune system and energize the individual. And it has been well recognized all over the world as agents that is being used like vitamins to relieve stress. What are the components in Jubeline? They include apigenin, litolidine, apigenin, litolin, and erigenin. All these chemical compounds are found in several series, like rice, maize, and even Jubeline, which is obtained from sorghum bicolumns. And all these compounds have been shown to readily cross the blood-brain barrier to exert several pharmacological activities. This is the owner of Jubeline. His name is Otumba Olajuwon Okumbena. He is the one that owns the product called Jubeline. And he has the recipes for the formulation of the product, which is not, I think, is not willing to release to the public. This study is based on the fact that, as I said before, stress is a very important factor in aggression, and Jubeline, as one of the indications, is for stress. Hence, we're trying to look at the possible effect of this product on stress. In fact, my laboratory has done a lot of studies on Jubeline because I'm doing behavioral pharmacology, looking at how drugs influence animal behaviors and the possibility of developing molecules that may be useful in the treatment of neurobehavioral disorders like schizophrenia and so on and so forth. So the source of animals from Animal House, University of Ibadan, and the drugs, Jubeline was obtained from the manufacturer, haloperidol, other drugs were used in the study. And the dose of Jubeline that were used for the study were obtained by previous studies, experiments, that is pilots that we did to get the right dose that we use for the study. And all the drugs were administered orally through gavage. The first one is to look at the model, which we well known as resident intruder-induced offensive aggression. This model is simply done by pairing the animals, a male and a female, just marrying them together in a cage for 3 weeks. After that, they were tested for aggression. Those animals that exhibit aggressive behavior were used for the studies, and we administered the various doses of the different drugs, as you can see in my slides. 30 minutes after drug treatment, the male animals were introduced, that is, male intruder were introduced to the cage of the residents' aggressive male animals after the removal of the females. And the confrontation between them was videotaped for a period of 30 minutes. Parameters assessed include latency to attack, the frequency of attack, aggressive posture, lateral threat, pursuit frequency, and so on and so forth. The other model is what we call isolation-induced offensive aggressive paradigms. In fact, it's so interesting that I was exposed to this sort of aggression just about 4 years ago when Professor Winner, who is the chief editor of Neuropharmacology in Europe, invited me to submit a paper for annual publication of the journal. Every year they have a special edition. So I was set into action. So can we study aggression in animals? So I went into literature and we saw a lot of models, out of which I'm describing now. So these animals were isolated. The male animals were isolated after weaning, and they were kept in isolation for 3 weeks. Mr. Chairman, behold, after 3 weeks, they exhibited serious aggression in contact with an intruder, which actually showed to us the importance of parental upbringing on animal behaviors. So just after establishing the aggressive behavior, for 2 to 3 weeks, isolated mice, they were treated by various drugs, including the drug of study, which is Jobelin. This is the video. Maybe I should escape the video for now. Apart from looking at the aspect of offensive aggression, we also look at the effect of Jobelin on defensive aggression, which is good for us, that in a life-threatening condition, we're able to defend ourselves. A drug that impairs defensive function is not a good drug, like the haloperidol-like drugs. Therefore, a drug that is devoid of defensive behavior should be a drug that we should pursue for the treatment of aggression. So this was done using resident-intruder paradigms. In this case, the intruders were the ones that were treated with the drug, not the residents. So that is the film. You can see me later. Data were analyzed using graph prism, and one way or another, followed by post-Harvard turkey test, was used. Results. Jobelin inhibit aggressive performance, but did not reduce the propensity to fight, as measured by the latency to first attack. That is, as you drop the intruder into the territory of the resident, immediately attacks. So that's what refers to the propensity to engage in fight. That was grossly reduced or impaired by haloperidol. So this is the result of the various parameters, the composite taken together. That is all what we are seeing can be put together as performance of aggressive act. Why this graph shows the latency to first attack, which you can see that you can see that is haloperidol, a typical antipsychotic drug used to treat schizophrenia. You can see how it impairs the the latency to attack. It prolongs it, which shows that this drug is reducing the propensity to fight, to aggression, and that is based on sedation. When an animal is sedated, of course, it will not be able to fight as it's supposed to, but Jobelin did not affect that. So the same picture is showing here, the same thing. This is just the effect of isolation, which is just like what we saw in the resident paradigms. So you can see from here that Jobelin did not impair the defensive behavior of the animals, which show that it should be a drug that we should pursue for the treatment of aggression. It shows that it has specific anti-aggressive effect, able to reduce aggression without impairing defensive mechanism. Conclusion, Mr. Chairman, and if you permit me, I can play the video clip for us. This study showed that Jobelin has anti-aggressive activities devoid of impairment of defensive mechanisms of the animal. And this property may encourage development as a remedy for aggressive disorders. However, further works are going on in my lab to look at the various compounds that have been shown to be present in juberlin, the most likely one that may be responsible for this effect. Thank you.
Thanks a lot.
Thank you very much. But to be consistent with the timing of the session, I would allow one question if anyone in the member in the audience has a question.
So any questions? Sorry. Yeah.
You look good in your dress. But you know, I worked with a lot of Nigerians at Harvard University of Washington, D.C. But you used to have a hat that is a cloth hat, you know. But this hat is different. This is also Nigerian, thank you. You must have purchased it at Hyderabad. Anyway, now my question is, animal data, when you translate it into the clinical trials, most of the time they do not fit in. That's why an area called, how much percentage you can give to your animal data that fit in the 80%, 90%? Any guess? No, let me repeat, let me rephrase it. How far your animal data is going to fit into clinical data? Exactly. That means in a translational research that we call it as, when you translate the results of animal models into the clinical trials, most of the drugs fail. That's the reasons why people, there is an area, emerging area called translational research. Okay? Now, how confident you are, how much percentage you can give your animal data is going to fit in?
Thank you very much. As I said, these models are predictive of human disorders. In drug development, as you know, we have a preclinical phase and then to the clinical phase. As you rightly said, we might not be expecting that the data from the animal studies should be almost the same thing. I mean, the 100% of beneficial effect in clinical settings. It might not be, but we might be lucky to get a better result, just as we had in the preclinical studies. These are models for drug development, and as I said, models could be predictive, and they have face validities. But as long as the data in the animal studies are promising, it suggests that we can take it to the clinical trials. It may be effective, it may not be effective. But that is the borderline, that is the truth. But they are all studies. And the end point of it is to see that the possibility of getting a molecule that may be useful in human disease condition. Thank you.
Thanks a lot.
Thank you, Dr. Solomon.